Okay, this is Minerva Biotechnologies, and today I'm going to be talking to you about a CAR T cell that we developed for solid tumor cancers. And this CAR T targets MUC1 star. So MUC1 star is very different from full-length MUC1. And that's an important distinction because you may already know that all of the therapeutics that have been tested against MUC1 full-length have all failed in human trials. So MUC1 biology has been poorly understood until very recently. So a lot of what's published in the literature has been proven to be wrong. So full-length MUC1 is comprised of a transmembrane domain, a cytoplasmic tail, and then it has a very bulky extracellular domain that is comprised mostly of these 21 amino acid tandem repeats. So that's what MUC1 looks like on a healthy cell, full length. On cancerous tissue, MUC1 is enzymatically cleaved, and this bulky tandem repeat domain is shed from the cell surface. And that leaves this little short transmembrane protein, and that is the growth factor receptor form. So there's an onco-embryonic growth factor called NME7. AB stands for the secreted form. And NME7 has two binding sites for MUC1 star. And those binding sites are ectopic, meaning they are hidden until MUC1 is cleaved and the tandem repeat domain is released. So NME7 has two binding sites for MUC1 star. Even at very low concentration, it dimerizes MUC1 star and it activates the MAP kinase signaling cascade. And so there are a couple of red herrings that have come up recently about MUC1. There are antibodies such as 5E5 and SM3 that bind to these trapped glycans. And these trapped glycans are said to be cancer specific, but they only occur on O-linked glycosylation sites. And the important point is that MUC1 star has no sites for O-linked glycosylation. So antibodies like 5E5 and SM3 cannot recognize the growth factor receptor form. We're also asked, what's the difference between MUC1 star and MUC1C? And the answer is there is no difference. Some of our competitors thought that they would be clever by renaming MUC1 star MUC1C, and they add a couple of amino acids onto the N-terminus. Now, this is a short list of all of the companies that have tried and failed to develop a therapeutic against full-length MUC1. And you can see they have all failed. They all target the tandem repeat domain, which MUC1 is cleaved on cancerous cells, and that tandem repeat domain is shed from the cell surface. The only exception is Genus Oncology had a synthetic peptide that bound to the it's a mimic of the cytoplasmic domain of MUC1. And you will understand that the cytoplasmic tail of MUC1 looks the same, whether it is MUC1 full length or this cleaved growth factor receptor form. So all of these have failed. So our therapeutic is a CAR T. And the question is, why haven't CAR T cells worked so well in solid tumor cancers? And the simple answer is there's no B cell equivalent in solid tumor cancers. And most of these blood cancers are B cell malignancies. And so the CAR T cells, the CARs bind to something that's on a B cell, like CD19. And you can wipe out the patient's entire B cell population, and maybe the patient will get the sniffles more often, but otherwise they're doing pretty well and they no longer have blood cancers. And it's a little bit more challenging on solid tumors. You can't just wipe out, one whole cell type. However, we're going to argue that MUC1 star is the B cell equivalent on solid tumors. MUC1 star is aberrantly expressed on over 75% of all solid tumors. And you can see it's expressed in really high percentages on these really killer cancers like breast cancer, ovarian, pancreatic cancers, lung cancers. And so the challenge was, MUC1 full length is expressed on all epithelial tissues. So how do you make a CAR T that's not going to hit your normal tissues? And the answer is you have to target MUC1 star, but you also have to target this hidden binding site, which is only exposed after MUC1 is cleaved and the tandem repeat domain is released from the cell surface. And importantly, our lead antibody competes with this Onco embryonic growth factor, NME7, for binding to this ectopic site. So this is a first-in-human clinical trial. It's no other therapeutic has ever been tested against MUC1 star. We have a diagnostic test, so eligibility is if the biopsy is at least 30% reactive with the antibody that is the targeting head of our CAR. The patients undergo standard Cy/Flu lymphodepletion. It's a standard 3x3 dose escalation. Right now, we're using autologous T cells, but this does not negate in the future we might go to allogeneic. And we have a very short manufacturing protocol that includes an antigen stimulation step. So our trial is being performed at the Fred Hutchinson Cancer Center. Lentigen produces our lentiviral vector, and Covance is our data patient data database manager. And I'll give you a very quick look at our first patients. And we had very robust CAR T cell expansion. The first patient had a grade 2 CRS. Unfortunately, this patient had her cancer progressed through 15 different lines of chemotherapy. She was very brave. Actually, she did very well on this. But during the 28-day evaluation period, she had a lung infection from a permanent catheter that she had in her lung. So she had to be dosed with steroids. And those steroids, dexamethasone, suppresses CAR T cell expansion. And so on the left, we're showing PCR of the CAR T cells. On the right, we're showing the absolute number of CAR T cells. And what you can see is that once the steroid treatment was stopped, then the CAR T cells started expanding again. And at day 28, she still had 8,700 CAR-positive T cells per ml of blood. And an early hint at efficacy, the CAR T cells destroyed 87% of her circulating tumor cells. Patient number 2 had very good CAR T cell expansion. She had also been heavily pretreated before the CAR T therapy. She has real good persistence of the CAR T cells, and we see at day 28, she had about 25,000 CAR-positive T cells per ml of blood, and at day 60, still 13,000. And remarkably, 155 days after her CAR T cell infusion, she still has a very significant number of CAR-positive T cells per ml of her blood. So we were slowed down by COVID-19, but here's a very quick summary, just broad analysis of what happened in our first couple of patients. We had no serious adverse events attributed to the CAR T therapy, no detectable organ or tissue toxicities, and we had very good CAR T cell expansion. And the early indicators of efficacy are the 87% reduction in the circulating tumor cells in the first patient. In the second patient, she had stable disease. And also by M-persist, she had a partial metabolic response. So a greater than 30% reduction in the bone metastases where most of her metastasis was. And that partial metabolic response persisted through day 60. Now I'll talk a little bit about Minerva's pipeline. And so our core expertise is really our deep understanding of the cancer cell biology and where that intersects with stem cell biology. So in this slide on the far left, you can see this is a breast cancer tissue. And you can see virtually every cell is just coated with MUC1 star. And we're probing this with our antibody C2, which is the antibody that we're in the clinic with. And you can see, unlike cancerous tissue, the healthy tissues are pretty clean so that we had minimal to no binding on healthy tissues. Now, if you look at the panel on the right, we're showing NME7, the Onco embryonic growth factor. And here we're showing a photograph of a day 3 human blastocyst. And you can see every cell is staining positive for the secreted form of the oncoembryonic growth factor NME7. By day 5, those cells are restricted to the inner cell mass. And by definition, the inner cell mass contains the cells, only those cells can become any cell in the human body. They completely self-replicate and they have no marks indicating cell fate decisions have been made. And on the far right part of this slide, you can see that all these cultured cancer cell lines are expressing NME7AB, the secreted form. So that goes from breast to lung to pancreatic cancers to prostate cancers. They're all expressing the growth factor, MUC1 star, and the oncoembryonic growth factor, NME7AB. So we have the CAR-T trial in the clinic right now against the growth factor receptor. And this is an example of our animal studies showing that the CAR-T cells eradicated the MUC1 star positive tumors in animals. And the more MUC1 star expressed by the tumor cells, the more efficient was the killing of the tumor. So that's MUC1 star, the growth factor receptor. We have this in the pipeline. We have an antibody against this oncoembryonic growth factor that should not be expressed past about day 10 of a developing blastocyst. However, we saw it in all these different cancer cell lines and in cancerous tissues. So here we're showing 2 mice. On the far left, we have 2 mice. One was injected into the tail vein with the breast cancer cells so breast cancer cells, and the 2 mice are totally metastatic by day 10. And if you look at this progression, you can see that in 4 days, this antibody has completely cleared up the established metastasis. Another thing in our pipeline is we have a small molecule that basically behaves very similarly. It prevents cancer metastasis. And this small molecule was identified in a screen that we developed where we said naive stem cells are a surrogate for metastatic cancers because they express MUC1 star receptor and they secrete NME7AB. So we looked at the naive stem cells and we looked for small molecules that kill off the naive stem cells, have no effect on the later stage primed state stem cells, no effect on fibroblasts, and those are the small molecules that inhibit tumor metastasis. So I've been talking about cancer so far. So now that we understand that these naive stem cells grow by NME7 and the MUC1 star receptor, we can make stem cells in this very early naive state. And what I'm showing here are stem cell-derived dopaminergic neurons. And we're staining for all the markers that you would expect to see on dopaminergic neurons. And the top row have been made from Minerva's naive stem cells, having been grown in NME7. The bottom row are primed state stem cells that were grown in E8 media. And then the bottom row was differentiated according to the Lorenz-Studer protocol. And the top row is the Minerva protocol that we did, an important improvement to Lorenz-Studer's protocol for making dopaminergic neurons. And in this slide, we're showing dopamine transporter protein, which is a very important protein. And we can see this at day 24. And you can see the top row, naive stem cells, the Minerva protocol, is superior to the bottom row in terms of the multi-center connectivities, the length of the neural projections, and so forth. So probably the most important thing is, are these cells producing dopamine, and how well? And we've also measured in vitro engraftment, but here I'm going to talk about the production of dopamine. And this is kind of a complicated slide, but if you look at the x-axis, we're plating different numbers of cells. On day 11 of this protocol, you move all the cells to a different surface. And so on day 11, we're plating either 200,000 cells per centimeter squared, 400,000, or 800,000. And all of the red bars show that it's the Minerva protocol that's being used. The blue bars are the Studer protocol. If it's a solid bar, then it used naive iPS cells. If it's a striped bar, it used prime state stem cells that were pluripotent, but sometimes embryonic and sometimes induced pluripotent stem cells. So what you can see right away, these are mostly day 60, but there are two bars there where we measured dopamine production at day 40. And what you can see is the more cells you have, the more dopamine is produced. And the takeaway is naive stem cells work better than prime state, and the Minerva protocol works better than the Studer protocol. And now we have an even further improvement of that protocol. If you look at the pink bar, you can see with our latest improvement, we have at day 40, the same amount of dopamine that is produced from 200,000 cells as was previously produced with 4 times that number of cells. So that's a significant improvement, and at an even earlier time point, we had over 40 nanograms per ml of dopamine being produced. So at this point, I'm going to say thank you very much, and I want to thank our collaborators at the Fred Hutchinson Cancer Center. Our PI there is Jennifer Specht, and the Minerva team. Thank you very much. We have a great team here, and our disclaimer is all Minerva employees hold stock options in our company, and we have contact information. Matt Britz is the Senior VP of Business Development, Ron Axelrod, COO, and importantly, he does investor relations. So thank you very much.